otherwise better and longer.
Obviously, in many regards, it's very well established, but this there are reasons, says Richard, why this is likely to change. So, over the course of this research, oncology has a
in many ways, it's all about that. I mean, the reason why it's actually whatever it is, something will spread on. The children's cancer, the reason why perhaps more relevant is that because it is a small field, it's also a strength. The community is much better at collaborating, it's much closer knit, it's much more willing to share ideas early in the application. And in many ways, the advances that we've seen in children's brain tumours, children's leukemia, particularly, which has really brought the genomics transcriptome revolution into cancer understanding and diagnostics. You know, it's sort of punching way of the best way, largely because of that collaborative effort that's sort of brought about by that close-knit community. So although it's perhaps not so well known, those of us in the field, it's always been a very established community, and in many ways, dare I say, a more mature community in the way our collaborates. However, what I would say is that the lessons that we're learning from children's cancer and the collaborative nature of children's cancer really spawning what could be very transformative understandings of adult cancers. And that's actually the heart of that is biology. So whereas when you look at breast or prostate or lung cancer, you're often studying the difficult situation of essentially a mature tissue within the context of a mature organ. So seeing the delta in biology between the cancer and the normal is difficult. The kind of diseases that we look at in children in cancer are essentially embryonic tissues, which have long since gone. And so studying the difference between that and a very abnormal structure in terms of its chemical abnormality, because it's normally an embryonic structure, allows you to see very big differences between the normal and the existing tumor. And that gives you a clearer insight into the underlying biology. I think what's fascinating is a lot of the biology we see in children's cancer, although it relates to aspects of embryogenesis, still the fundamental biological processes hold true in adult cancer. So that clarity we can get by studying children's cancer is something which I think can teach us a lot about adult cancer. The reason I mention that is that the children's cancers are very different from adult cancers, no doubt, but actually as our ability to use larger data sets, particularly deep learning approaches to genomics, transcriptomics, and histology, as we get a holistic view of that disease in those settings, we can really start to bring those fields together and understanding how adult can teach about pediatric. And that, for me, that's the exciting frontier, is how we look at the entire development system and understand how cancer develops, rather than say in a quite binary way, this is children's cancer and this is adult cancer, and never this way to meet. I think the exciting frontier will how we bring those together.
Yeah, genuinely interesting, that, isn't it? And it really hones in on this issue of boundaries between scientific disciplines that are often, in effect, artificial and probably not always that helpful. So that's a lovely snapshot of where we are now. But what about the last 20 years? How has Richard seen his field and his work change in that time?
20 years ago, if you look back at children's brain tumours, which is the area where I've worked most, children were regarded, with brain tumours regarded almost as single entities. That diagnosis was made by looking down the microscope, looking at the morphology of cells and saying, this is cancer X, Y, or Z. And then the children were grouped into those broad categories, and they were treated on the basis of that. And obviously that's very crude, poorly defined. Now, because of the rapidly over the last 20, 30 years. We've been able to understand where these cancers are formed, why they are distinctly different, and then why there are different forms of these tumours and how to treat them differently. And that's had real practical impact. So the World Health Organization now classifies children's brain tumours based on the genetics and the genomics, one of which Parag has been involved in, and it's also leading to new treatments. But from my own personal perspective, what's been amazing is the sort of journey of going through children's cancer and seeing how origins of cancer can dictate clinical outcome. The sort of, I guess, eureka moment for me is, well, why not all kinds of cancers as well? And so my lab, increasingly half of my lab, focuses on cancer as a continuum from birth to old age, and how does cancer arise in the context of development, which we continue to develop throughout our lives. And most recently, that's such a very exciting discovery about metastasis, which is just about to be published. So those kind of insights that when I started my career 30 years ago, I never thought I would work on metastasis around cancer, and I'm doing that, and actually insights of children's cancer.
Now, another really fabulous example of change over the last 20 years is some spectacular work from Richard's lab, which delved into the biology of the different subtypes of medulloblastoma. The clinical benefits of which really could be something special indeed.
So we knew 20 years ago that if we took 100 children with medulloblastoma and gave them a classic radiotherapy or surgery radiotherapy, chemotherapy, that about 20, 30 years ago, about 60% of them would survive and 40% would die. We had no idea who it was going to be. I mean, crude measures like metastasis diagnosis, like in all cancers, was always bad. But pound for pound, if you like, if you took one child who had similar looking disease in terms of stage, you couldn't really predict what the outcome would be. We had very vague understanding. The first step was, the use of transcriptomics, which my lab and many other labs around the world were involved in. And the early sharing of those ideas was very important to set the classification. Essentially, we think about them as only 4 subtypes. And one subtype of that was noticed early on did particularly well, even if those children were metastatic, which was quite phenomenal, perhaps before the WIN subtype in the gene mutations. But the real question was, why do they do well? Is it because they are, they're just not bad disease, or they don't proliferate as much, or they die quicker, or they're more really sensitive? And I think one of the first steps forward with development of biology for us was when Paul Gibson was a postdoc in my lab at St. Jude, and we mapped the transcriptome of the human tumours onto the developing mouse brain. And what was really fascinating was to see that they actually had a completely different origin than had previously been thought. So they were born in a different place. So the next step was, okay, they're fundamentally different from the start. That kind of makes sense, but still doesn't explain why they do better. And then a second postdoc, Wellington Phoenix, actually came to my lab with the idea. He just worked with Sally Temple on the blood-brain barrier and relationships with neural stem cells. And Tim had got the hypothesis that because Wnt signaling was critical to maintain the blood-brain barrier, then actually because these tumours try to inhibit Wnt signaling because they had it so active, that would have an effect on nearby blood-brain barrier and shut silence it. He was absolutely correct. He published that in Equal Paper in Cancer Cell. And the reason that was important to go back to the clinic is that he demonstrated actually, because there was no blood-brain barrier, those children have about 1,000-fold more chemotherapy in their tumours than a child that has an intact blood-brain barrier. So that gave us a reason why those children would do better. Now, the really exciting part of Tim's work going forward with work that now being Undefined Cancer Research UK is actually to understand, okay, how do we treat those children better without any radiotherapy? And we noticed that those tumours are absolutely plagued with antibody from the systemic circulation, which is unusual because there's no blood-brain barrier. So we're now working on basically an antibody which will target specifically proteins on the surface of the medulloblastoma that would carry a payload of the toxin. So theoretically, we could just give that antibody to those children and they would never need radiographic vaccine or surgery, which would be really exciting because there's a big group of children, about 15% of kids with medullome. There are disproportionate numbers of damaged survivors because they do well. So that will be tremendously exciting as a way of giving a completely new treatment for children.
Really could be an incredible example of how biological understanding feeds right through to clinical impact. Now, I will put some links to this work and some more materials on the WNT or WIMP subtype of medulloblastoma in the show notes, so keep your eyes peeled for that. Also, keep an eye out for an upcoming article that we've got planned on the development of the technology that Richard mentioned there. That's the payload carrying antibody. That should appear on Cancer News soon, so keep a look out. But for now, back to the issue at hand. And there's an interesting aspect of all research on colorectal cancers, and that's the importance that's placed on the side effects of any potential therapies.
Yeah, and there's two reasons why side effects are very important in children. One is that children are developing, obviously, and so there are fundamental challenges. For example, in the developing brain where I work in, if you're raising a developing brain, the consequence for the child in terms of intellectual and endocrine outcome is so bad that it's worse than the cure. So that limits what you can do. There is another reality, and is that if you cure a child at the age of, say, 6, hopefully they've got another 70 years to live. And thanks to the work of organisations like NG that I used to work in the States, that have really followed children very carefully over now decades, it's very clear that children who have been treated for cancer in the past with chemotherapy, radiotherapy, have very short lives. I mean, these children run into problems with cardiac disease and other disease at the age of 30 or 40. So the long-term effects of cure are very, very significant. Now, of course, if you give a child an extra 20, 30 years of life, that's a wonderful thing. But if we can do that without causing that damage, that's an even better thing. And what's interesting is, again, I think this is another lesson for medicine, is that as we have much bigger data to our disposal and we're actually able to join fields to deep learning approaches, I think the other exciting frontier will be how we blur the boundaries between disciplines. So if we look at the long-term effects of risk from cancer or treatment, it bumps into cardiology and respiratory and gastrointestinal. And I think those disciplines can also learn from the side effects that cancers produce, but also the long-term risk of those diseases, because ultimately the risk factors of cancer are shared in cardiovascular disease and cerebrovascular disease and neurological disease. And so learning about holistic health and how we can pathways to prevent and intervene early needs to go beyond cancer into the whole aspects of health, and that's a really exciting opportunity.
Yes, exciting indeed, and it leads beautifully on to the next thing I wanted to talk about with Richard, which was where next? What will we see develop in the next 20 years?
Yeah, I mean, I think obviously we could talk about granular examples of individual diseases like children's cancer I work on or other cancer types. I think the big challenge for medicine generally, and I would like to stick with that, is the notion that all of medicine, modern medicine, is predicated on the idea that we have disciplines. So we have cardiovascular, we have gastrointestinal, we have hematology, we have pediatric oncology. And that's valuable because everyone can be an expert in their individual spaces. But one thing that COEK has been a cheerleader of, as other organisations, is early detection. And so as you think about early detection of disease and intervening early, it makes no sense to think about individual diseases specifically, because the risk factors we know for cancer are shared by other disease types. And that's largely because of the insults that we have to produce cancer are also going to cause other tissue damage or cause other kinds of abnormalities. Cirrhosis, for example, and lung damage, for example, smoking with chronic obstructive airways disease, but also lung cancer. You can list them all. And so what I'd love to see is an increasing understanding of how risk factors, both in terms of inherited risk factors and genetics, but also in terms of exposures and different geographic locations, whatever it might be, and how your body responds to those over time, how that puts you with early intervention on particular pathways of healthcare. So we start thinking about the ridiculous situation where you eventually have your heart attack in Sainsbury's, or you find a lump in your breast, or whatever it might be. But actually, early on, we know this person has, for whatever reason, risk factors in this pathway. So you would have a tailored pathway which is dealing with all your health, whether it's cardiovascular risk, and respiratory risk, allergy risk, as well as dementia risk in the future. And I think we'll increasingly realise that there's an overlap between exogenous risk factors as well as intrinsic risk factors and the kind of health pathways we go down. And it's more than just doing a better job of treating cancer early. We know, for example, that obviously the NHS model, which is a good one, is keep people as long as possible and as well as possible, and then they die more quickly when they're 80, 90, whatever. And that's great because it's great for them to enjoy long health, but also it's much less costly than people going downhill gradually. But we've also recognised that individuals, I think increasingly we'll see this, if you intervene early, say for cancer, and are successful at treating that, actually what may well happen is they die at the same point in time of another disease. So you've not actually increased their lifespan, and you may not have actually increased their health. You've just stopped them getting cancer. And so that holistic view, I think, will also help us with that vision of keeping people as well as possible, as long as possible, as cheaply as possible, which frankly is important.
Okay, time to briefly change tack and talk about what it's like to be a researcher. How has Richard seen that change over the last 20 years?
Yeah, I think that's a difficult one, because I think everybody, it's easy to moan about things, and it's easy to say that, oh, it was always better in my day, kind of thing, and I don't think it's changed that much, to be honest. I think, and that's because people don't change that much. So we like to sort of pontificate about, the purity or I don't know, the higher mind level of research, but it's all people working in an organisation, and people's fundamental motivations don't change. I think that there is a better level of collaboration than before, but I'm spoiled working in the paediatric community where collaboration is just in our nature. And I don't think that's because we're any better people, it's just by necessity that's happened. I do think one of the biggest challenges is, and everyone talks about it, is data. And I think that, I don't say that flippantly, but our capacity to generate data has massively outstripped our ability to analyse it. And so we've been putting a lot of thought here in Cambridge about how we don't become, data sausage machines where you just generate data for the sake of it, and then after that think, what do you do with it now? Actually, how do you use this data in real time so that you're not
Hello and welcome to Cancer Research Matters, the new podcast from Cancer Research UK aimed at our research community. My name is Phil Prime, and for each episode of this first series, we'll be speaking to some of the fantastic researchers behind the work we do. Because this year the charity is 20 years old, what better time to take stock and consider where we've been and where we go next?
This has become a lot bigger than I thought. It's become a lot more exciting than I thought. We've learned a lot more about lung cancer than I thought we would.
That was the voice of Professor Charles Swanton. Now, you might know Charlie as our chief clinician, but of course his day job sees him firmly rooted in the research based at his laboratory in the Francis Crick Institute. His work has involved asking some really tough questions around how lung cancer cells evolve, spread and become resistant to therapy. Now, in many ways, the kind of culmination of his work on this clonal evolution aspect to lung cancer has been the Tracer X project. And as you just heard from that soundbite, this really has been a groundbreaking project in so many ways. But before we get to that, it's time to wind the clock back with Charlie and find out how he ended up in research. In the first place, Charlie started his training in medicine and is now, of course, a clinician scientist. So why the pivot to research?
I think it was due to the fact that in my end of my first year, beginning of my second year at medical school, my father was diagnosed with a cancer. He didn't know what it was at the time. And it was a pretty difficult time for the family, as you can imagine. I was very close to my dad. And he had this sacral tumour at the bottom of his spine. And to cut a long story short, after a series of investigations, some of which probably wouldn't have happened had one or two doctors not insisted on it, he was diagnosed with a lymphoma, high-grade B-cell lymphoma, and had chemo first, then radiotherapy. And 20 or 25 years later, he's just retired from the NHS as a consultant cardiologist. And I think that illustrated to me at the time that somebody's research at some point in medical history had made a difference to my father, and that was really truly inspirational, because I knew full well that without that combination of chemo and without that radiotherapy, my dad wouldn't be with us today. Really from then on, I thought, this is something I want to be involved with, and I wanted to spend the rest of my life doing it.
Always worthy of note, I think, the sheer number of cancer researchers that have personal reasons for doing the work they do. It really does make you a very special bunch indeed, I think. So, having gone into research, why did Charlie end up focusing on lung cancer?
It's a confluence of opportunity, really. I initially was a breast cancer doctor at the Royal Marsden, which I enjoyed very much. And when I became a consultant, I worked with colleagues at RMH, which was fabulous, incredibly rewarding. But I also recognised it was quite a crowded field, breast cancer. It was a field in which a lot of major discoveries had been made back up to sort of 2008, 2009. That's not to say that more amazing discoveries hadn't been made in the last 14 years, because of course they have. But I sort of recognised that sort of fitting in as a sort of junior clinical academic into what was a pretty busy field would be tricky. And around about that time, after about 3 years of being a consultant, like 2011, this opportunity came up at UCL, in part because of CRUK's investments in thoracic oncology and lung cancer research. I made a Cancer Research UK chair available through the centre, the UCL Centre. And it had been open for 18 to 24 months. I think they advertised and nobody had shown any interest, because people weren't terribly interested in lung cancer back in the early 2000s. But of course, with precision oncology and EGFR inhibitors and what have you, prior to the advent of checkpoint inhibitors in lung cancer, bit by bit, enthusiasm for lung cancer specialty increased, and almost as soon as I joined, I think you could sort of feel the excitement and enthusiasm for this specialty that had previously been sort of a specialty that people didn't want to go into because
Hey, there goes my door.
advent of precision oncology and EGFR targeted therapies, amongst others, the practice of thoracic oncology became much more rewarding, and the outcomes of patients, these subsets of patients, became very much better. And so I was fortunate enough to be offered this job in 2011, and I've never really looked back.
Yeah, so some interesting hints there as to how the field of lung cancer research has shifted over the last 20 years. So what else has changed during Charlie's time?
The general interest and investment, and that's not just in the UK, considering it being the major leaders in this field of driving these cancers of unmet need, which I think has made and is making a real difference. But also globally, there are many more lung cancer charities than there were 15 years ago. I think people now recognise lung cancer as a disease that's worthy of huge investment. I think a lot of the stigma of lung cancer has gone, and I don't think it's completely disappeared. And as a patient once said to me, it's actually doctors who are responsible for the stigma, not patients. I think that's a point worth thinking about. What does stigma mean? It's this idea that most lung cancers today are still caused by smoking, and we have to recognise that smoking is highly addictive, and we have to recognise that tobacco control could not be more important, and that the consequences of tobacco exposure for patients can be horrific, and we have to do all we can to control tobacco use and to help our patients through this diagnosis. And I think there's a recognition now that this is a disease, as I said, that's worthy of investment and desperately crying out for more research. And there's a sort of renewed enthusiasm for research, all the way from the bench, the lab to the bedside. And I think the last 5 years particularly has seen some tremendous advances in our understanding of lung cancer, advances therapeutically in patients. And of course, checkpoint inhibitor therapy would be central to those advances, in addition to the precision medicine I mentioned earlier. So, we're now looking at some long-term survival outcome in thoracic oncology, around about maybe 50, perhaps even 20%. We'll see. You might expect to see long-term survival. These data has been brought into the sort of early disease setting, both through fields after surgical resection, but also neoadjuvantly before surgery to shrink the tumour. And there's very exciting data now showing the efficacy of neoadjuvant checkpoint therapy in certain tumours, which is really very, very exciting. It's going to change the way we manage patients with this disease of lung cancer over the next 5 years, and much more neoadjuvant therapy, I think, we've ever done before. Because you've got the tumour inside you, covering the T cells against the tumour in response to checkpoint inhibitor therapy, and that's probably why neoadjuvant therapy is looking so exciting. So there's lots of hope is the short answer. But also there's a lot of enthusiasm around early detection and a field that I'm increasingly interested in, prevention.
Clearly, the last 20 years has seen lung cancer as a field become incredibly interesting and exciting for researchers. But what about the minutiae and the day-to-day of actually doing the science? How has the way as a researcher changed?
That's a good question. I think a lot has changed in the last decade. I think the first thing that's changed is the recognition that some of the problems we're facing are so large that they can only be addressed across multiple labs, and hence the need for team science, collaborative science. That's catalysed new programs, including Cancer Grand Challenges, that enable funding to move across borders in a collaboration between Cancer Research UK and the NCI. That's led to several very large collaborative grant programs in disease areas that had been previously overlooked because they were considered to be too large to tackle with a single program grant. So I think we're looking at grants that have been mobilised to fund the big questions, a recognition from our funders that team and collaborative science is important. I think the recognition that it's not only about publishing in Cell Science and Nature, it's the quality of the science, it's the impact that science has on mankind that's important, not necessarily where it's published. And I think the other thing is commercialisation. I think 10, 15 years ago, spin out companies would be very unusual from a group leader, PI in the UK. But I think partly due to the fact that money has been so cheap over the last decade and interest rates have been so low, this has, I think, catalysed a lot of sort of high-risk investments in research that may have a future major clinical impact, and has led to several biotech companies being spun out, particularly in the last 5 years. And a recognition from our funders and our university bosses that this is a laudable achievement, that this is as important as publishing in high quality journals, that you or publishing high quality science, that spinning up a company is probably the fastest way, or arguably one of the fastest ways of getting a scientific discovery into a sort of therapeutically tractable approach for patients. That's very hard to do in the academic sector and requires considerable investment when you're thinking about clinical trials and what have you. And of course the risk is very high. And taken together really that lends itself to commercialisation and biotechnology spin-outs. And we've seen many of these companies in the last decade spun out of CRO funded research, which was, I think, great to see.
Okay, now the beginning of this podcast, you were promised Charlie's view on TracerX, and we will. But before we do, I just really wanted to dig into this translation. CRUK's new brand, Cancer Research Horizons, is of course set up to carry on the tremendous work that the charity does to essentially push this process along. So I wanted to ask Charlie, what does it mean for this generation? After the early career gives more pride to people who are familiar.
Without a doubt, and also, but it's not just it is a generational thing, but it spans generations. Because when I was doing my PhD back in the mid-90s, in general, it was considered as going to about time spinning out a company, in amongst academic circles. Now that is absolutely not the case. It's quite the opposite. It is very much encouraged and supported, and so that's supported from the top down. So it's not just the juniors, postdocs and PhD students wanting to work, being more entrepreneurial, which of course they are, which is fabulous to see, but they're encouraged to be entrepreneurial by the funders and by scientific leaders who run the institutes, which is great to see. But we shouldn't neglect the bedrock of all of this, which is the scientific discovery that's enabled the translation. This is the fuel for the translational engine. Without the discoveries, there's nothing to translate. So we have to keep investing in the nuts and bolts and the mechanisms of biology and nature, essentially. So I think there's a sort of virtuous economic cycle here too, if you think about it. We spun out a company called Achilles Therapeutics 3-4 years ago now. And that company now employs 200 people in the life sciences industry in West London. So this is providing scientific jobs and experience for postdocs leading laboratories in the UK and elsewhere to experience life in biotech. Now, we don't know what the future holds in this field, and I think the point is that they've got the experience they've got, regardless of the success or not of Achilles. They can take that with them into other biotech companies that have in the UK or elsewhere. And of course, if one of these biotech companies spun out by CRUK, MLC, Local Trust, whoever, universities, AGIs in the UK, succeeds, that the potential revenue stream back to the funders or the universities or research institutes to fund further discovery research is incredible. The most important aspect of this is that you're talking about generating diagnostics and medicines for patients.
Now, if that's got your entrepreneurial juices flowing, and I can't imagine why you wouldn't have one, then I will put some links in the description to this podcast as to how Cancer Research Horizons can help you with your translational journey. Okay, as billed, it's time to turn our attention to TracerX, or tracking cancer evolution through therapy. First things first, what exactly was the project?
So TracerX started back in 2014. It was a CRUK funded program. I think it was CRUK's largest investment ever in lung cancer. And the plan was to recruit approximately 800 patients, early stage disease. That's a primary disease that hasn't spread beyond the chest, that was surgically operable. And we consent patients for entry into this program. It's not a trial, it's a longitudinal observation study. And patients have their tumours resected as part of standard of care. And then they're sent to us for what we call multi-region sequencing to understand how the tumour evolved from a single cell to a cell with hundreds of millions, if not billions of tumour cells in. And ultimately metastasize. So if tumours recur, we'll ask for a biopsy of that recurring tumour to understand how the metastatic disease that had spread beyond the chest was related to the underlying primary diagnosed months and years before. Now, we follow all patients, all 800 patients in the study, including those that don't relapse, that are cured. So that way we can compare the genomes of patients who've been cured, patients whose tumours have relapsed. And that enables us to start to make some inferences about the evolution of tumours that are potentially born to be bad compared to tumours with a better outcome, and start to infer the evolutionary patterns across these tumours and what it is that seeds a metastatic competent subclone, that is, a small group of cells in the primary tumour that ultimately has the capacity to spread to distant sites of disease. And we're applying an array of analytical techniques to the data, and there are several exciting endpoints. The primary endpoint of the study was to ask, is there a relationship between the genetic diversity of the cancer and outcome? To which the answer is we think there is, and the more chromosomally unstable the tumour, the worse the outcome. And then we've also been using blood from patients that kindly donated the blood to look for circulating tumour DNA hallmarks of the tumour coming back after surgery without evidence of the tumour being on the CT scan. And we call that circulating tumour DNA minimal residual disease technologies, and we've been working very hard developing those technologies for implementation, and a couple of assays have been derived from TraceRx in partnership with industry, showing that these clinical utility
Please gonna need another course.
biomarkers in the US and currently in use in clinical trials. And then, of course, we're very interested in the immune system, how the immune system shapes the evolutionary biology of the disease and ultimately may prevent tumours from metastasizing, or in a worst case scenario, the tumours may escape immune control and metastasize, and we're learning more than ever about that process now. And that's led to our understanding of what's important for immune recognition, which include these truncal neoantigens, so these proteins that are in the trunk of the genes which we present in every tumour cell. And those are the ones that we are going for in a targeted therapy approach with immune therapy, where we will ask the T cells to recognize specific truncal neoantigens and hopefully larger numbers to shrink the tumour. And then we've also found that those truncal neoantigens
Until when? Oh god.
so they're clearly
Um,
and through a series of experimental analyses over the last 15 years, got to the stage where the process finished in pretty much. We recruited patients, and milestones reports will be published soon.
Okay, so you get a sense of how ambitious this project was. But generally speaking, what have we found out so far? What do we know now that we didn't know at the beginning of TraceRx?
So I think, first of all, I think we've added evidence to the debate around genetic diversity now. So the more genetic diversity, the worse. I think the discovery of truncal clonal neoantigens from TraceRx, defining genes and leading to the spin-up company that I mentioned, Kidney Therapeutics, that will take adopted T-cell therapies from the bench to the bedside. I think understanding immune predation in untreated, treatment-naive tumours at a resolution that's never been done before has been tremendously exciting, showing the different ways in which tumours can evade T-cell mediated predation, including HLA loss of heterozygosity, antigen presentation machinery disruption, neoantigen copy number loss through chromosome instability, which has shed light on the role of chromosome instability more directly in the context of allowing immune evasion to occur. And then I think in TraceRx, using the high resolution data to define selection in untreated tumours, which we've shown now more clearly than ever, I think, and the role that plays in the metastatic process and the emergent metastasizing subclone. And then last but not least, I think the MRD technologies have been very important, using
I reckon most of the time is gonna be spent working on Shieldy like.
ctDNA panels. There was a lot of resistance to this, but subsequently we've shown that it's a pretty sensitive way of detecting recurring tumours in blood before there's evidence of disease on the CT scan, and that's now enabled us to work with pharmaceutical companies and how to set up adjuvant trials, not us but also the community to set up adjuvant trials using these panels that we co-developed with Natera and Archer, and now Invitae, that have got FDA breakthrough device approval, using these biomarkers to better segregate high-risk and low-risk patients in the adjuvant setting. So I think that those would be the main findings. There are going to be others too that I can't talk about today, but I think those are the main ones.
So it's clear that the findings come out of the TracerX project already have nudged the dial on our understanding of lung cancer. But the thing about TracerX is even though it started off as an already pretty ambitious project, that ambition has just grown and grown as it's gone on. And if you think of the project as at its core an incredible data generation exercise, you begin to see why.
We'll probably be analysing this data for another decade, at least, I suspect. It wouldn't surprise me if there are TracerX papers 10 years from now. This has become a lot bigger than I ever thought it would be. It's become a lot more exciting than I ever thought it would be. We've learned a lot more about lung cancer than I ever thought we would. But most importantly, we've trained principal investigators in lung cancer and other tumour types from TracerX, who are now running their own groups, getting research professorships, winning prizes for their work, that have been trained through the TracerX stable, as it were. And I'm most proud about that. We've got phenomenal scientists who come through the group who are, now just trailblazing in their own disease areas and just doing absolutely brilliantly. And it's just such a joy to watch that. And we've had over 15 junior postdocs who came through TracerX, or PhD students who are now principal investigators, either in the CRIC or outside the CRIC at UCL or other universities across the UK, but also globally, interested and working on cancer evolution and lung cancer. And so this massively expands the opportunity to break through barriers of our understanding of the disease, because we've got so many more talented people working in this area. And that's great, I think, ultimately for patients and for our understanding of biology and cancer. That to me is the best thing.
So I think for early career researchers, that's probably really lovely to hear, actually. A reminder that their academic journey in what is a flagship scientific project like this is so important. Also, I think it's testament to the power of big data projects like this that are collected, curated, and tended with care and talent, that unexpected collaborations and studies can come out of it.
One of the joys about TracerX is it's opened up avenues I would never have got interested in. So it's chromosomal DNA, cancer cachexia with Mariam Jemalhanjani is leading that team now. She's spinning out her own group in the cachexia field. We've got Julian Downward, George Cassiotis working in the fields of signal transduction, endogenous retrovirus envelope recognition, therapy response. We've got Eric Tsai working on the tumor microenvironments in TracerX and applying mathematical modelling to our understanding of clonal evolution. Obviously, we've got Nikki Canavan. Nikki's really getting into the depths and weeds of understanding of evolution at subclonal level to understand the selection processes that are occurring as tumors evolve. Kevin Litchfield's working on the cancer microbiome in lung cancer. And we've got, as part of our collaborative team, bringing in all this area of expertise, epidemiologists, stem cell biologists, Ilari Melanchi and others. We've got interest in stem cell biology and cancer metastasis working with us on evolution and cancer initiation. So and of course there's the early detection work as well. So we're really covering the whole spectrum of the CRIC at UCL and Manchester, from early detection, prevention, through to adjuvant therapy, minimal residual disease trials, and late stage disease and our understanding of the metastatic process, really from the first cell through to the last cell.
So, what's next? Well, this ambition is clearly contagious because despite the ongoing data analysis and discoveries which will come from TracerX, Charlie and the team are now thinking about the next phase. Whilst it's too early to give any real details, TracerX Evo will be a project that attempts to get some very big answers indeed.
We're still scraping the surface in our understanding of evolution. We know next to nothing about evolution at the single cell level. We know next to nothing about the initiation of cancers, which will hopefully lead to a whole new suite of approaches to prevent blood cancer. I'm specifically alluding to our recent interest in air pollution and lung cancer and trying to understand how the sort of first stem cell progenitor arises from a pollution-exposed lung. I think that's going to be a critical part of our development program over the next 5 years. Understanding how this first nascent stem cell exists, how it's changed from a normal epithelial cell in the lung tissue to become a cell with a progenitor-like state, I think it's going to be crucial to understand. And then as we progress, I think we've still got a lot of work to do on these immunological disease assays. We've got more biology to understand, sampling, time points, etc. CTC technologies are advancing all the time, so I think tumor cell technologies are advancing all the time. These are ability to understand genomics at a single cell level, which will hopefully inform risk in the context of circulating tumor cells that are present in the blood after tumor resection before relapse. And then really sort of a sampling issue. We still aren't sampling these tumors sufficiently, and I think there's an unmet need to include autopsies in any analytical program in cancer evolution. So I think that's something we're going to be focusing on very much in the next 10 years. So that, what we found in TRACERx is the more biopsies you look at, the more heterogeneous, obviously, and the more polyclonal the metastatic disease process is. So sampling one biopsy is just simply sampling one metastatic site simply insufficient to be able to infer the degree of complexity of the metastatic process. And then tying in all of this to our understanding of the microenvironment is going to be crucial. So we need to bring more microenvironment people into the team and understand the macroenvironment. How does the cancer impact the host? And that's where the cachexia work is going to be so crucial. What are the signals between the tumour and the normal tissue that signal your muscle to start to catabolize itself, or your body fat to start wasting away? How is that signal mediated from the tumour? And is it required for tumour growth? Are the metabolic products of muscle breakdown and fat breakdown required for tumour growth? And this is part of the process in this sinister syndrome called cancer cachexia, which I think is so important. So I think those are some of the areas that will form the mainstay of our broader TRACERx consortium focus over the next decade.
Now, just before I leave the final words to Charlie, just time for me to say thank you so much for listening. Please do have a look at the show notes for links to articles about TRACERx and some of Charlie's work on air pollution. But for now, time to get a snapshot of Charlie's long-term hopes for cancer research. If you could know one thing in 20 years' time that we don't know now, what would that be?
I would like to know, more than anything, how cancer cells communicate with a complex microenvironment, and whether clones operate together or they're completely independent. And I think that degree of crosstalk between the cancer cell and its community that surrounds it is going to be crucial to our understanding of the process of cancer dissemination, but more importantly, will inform how we disrupt it and ultimately target it for patient benefit.
Hello and welcome to Cancer Research Matters, the new podcast from Cancer Research UK aimed at our research community. My name is Phil Prime, and for each episode of this first series, we'll be speaking to some of the fantastic researchers behind the work we do. Because this year the charity is 20 years old, what better time to take stock and consider where we've been and where we go next.
The junior group leaders that we recruit are in that optimal creative time of their career, and actually many of them would have great ideas for spin-outs, but it's a bigger risk for them.
That's the voice of Dr. Simon, senior group leader at the CRIC, and he's also an honorary professor at UCL. His work is largely focused around DNA damage response. He also happens to be wonderfully entrepreneurial. and was a major player in the spin-out Artios Pharma, which, as you might imagine, focuses on DNA damage response and possible ways of utilising that in therapeutics. And if all that wasn't quite enough, he's also the director of COUK RadNet City of London. For those that don't know, RadNet is a network of facilities and expertise which has been set up to take on major challenges in radiobiology. So, what that means for us is that Simon is just a great chap to speak to about all aspects of cancer research, how it's changed over the years and what direction it might go in the future. But before all that, it's time to find out how Simon found himself at the sharp end of genome stability in cancer.
It was all a bit of serendipity, to be honest with you. I was in Edinburgh doing my undergraduate. I actually went to do chemistry, but I was fortunate because it's a 4-year degree for English people going to Scotland, and I was given the opportunity to study a variety of different topics around chemistry. I did genetics, I did molecular biology, biochemistry, and the exposure to molecular biology, well, and genetics as well, just fascinated me. You know, it was cutting edge, whereas I felt I suppose wrongly now in hindsight that chemistry was a little bit dated. Certainly what we were doing at the time was, you know, seemingly discovered 100 years ago, whereas molecular biology was all about, you know, the advent of genetic engineering, and I became very excited by that. And actually I approached the lecturers and said, look, you know, is there a possibility that I can switch or change to molecular biology? And that was an important decision. One of my lecturers in Edinburgh, Jean Beggs, an outstanding scientist, she and I, she walked into the lift that I was getting to leave the building, and she said to me, so what do you fancy doing? You know, I said, well, I'm excited by all sorts of things. I want to do a PhD, but I'm not quite sure where to go. And she said, oh, you should contact this guy. He's just come back from Berkeley, where he's done his postdoc. He used to be a PhD student in my lab. You should check him out. And that was Steve Jackson. And so that chance meeting in the lift was a career-defining moment for me. I remind Jean occasionally when I see her of that. And I went to Steve's lab, and I suppose the rest is history.
Okay, so Steve Jackson. As many of you will know, Steve, over the course of his career, has been one of the leading minds of DNA damage repair. And in fact, in case you haven't heard it, we had him on the podcast earlier in the year, and I'll put a link in the description. Please do check it out if you haven't done already. So, that is how I think that these would have been formative years for Simon's career.
It was an amazing place to be. You know, we had a really amazing atmosphere. I think it wasn't clear initially because when I started, there was only a small number of us in the lab, but they made this really key discovery around DNA-dependent protein kinase, DNA-PK. And this was like the first, I suppose, of the stress response kinases that was identified with respect to DNA damage response. And Steve had some seminal papers. So that was a real breakthrough for the lab, and we were suddenly inundated with loads of people applying, and the lab grew exponentially. We had people working in the equipment rooms, and it was a crazy time, but it was a really special time. Towards the end of my PhD, of course, Steve came up with this idea of trying to develop therapeutics against DNA repair proteins, and to be honest, it met with a little bit of skepticism at the time because people didn't really understand the opportunity. I'm not sure it was totally clear, you know, why would that work, right? You know, surely you just get toxicity if you're combining these inhibitors with radiation or what have you. And I don't think it was not really clear at the time, but Steve managed, I think it was a difficult journey, but he managed to persuade investors. So during this time, I obviously disappeared off to do postdocs in Boston, at Dana-Farber at Mass General Hospital Cancer Center, and that was a great time. And then I came back to the world famous Clare Hall Laboratory, headed by Thomas Lindahl, and it was the most amazing place, surrounded by some of the very best scientists in DNA around the world. Thomas went on to cell cycle, and everybody else there was just spectacular. I was incredibly naive. I was scared myself. I had a lot of support, particularly from Steve Weston, my mentor and good friend for 20 years. I liked to be next to each other for that time. And, we started in C. elegans, little worm, the first Manitoba organisms. And we discovered the BRCA genes in C. elegans. We discovered the Fanconi pathway. We started to exploit the genetic system to try and understand more about that pathway. And weeks would take a year in maths. And so there was a real benefit there, particularly around reducing the numbers of animals. This was a very, good system for using model systems. But of course, that works, and they obviously have strengths, but they have weaknesses, as every model system is the case. There's nothing perfect, unfortunately. But that was I learned very quickly of the need to adapt and to evolve, and so I continue to work in C. elegans to exploit that system, but also to complement that with generating mouse model informed based on the genetics that we discovered in C. elegans. We started to become trained in biochemistry, so we could start doing mechanistic stuff. Then we started to do a lot of human cell culture work, and more recently we've become adept to single molecule biophysics, where we can now monitor proteins at a single molecule level in a dynamic environment, and that's been a real breakthrough for us in terms of understanding mechanism.
So, chemistry to nematode worms, quite a mix of individual protein molecules, not to mention an incredibly exciting time in the burgeoning field of DNA damage repair. All of this from a chance meeting in a lift. So, once again, poking its nose into the scientific endeavor. Now, one thing that I think it's safe to say has changed over the last 20 years is the awareness of academics of the avenues to translate the work. And Simon's journey really does show why it's important to understand that from early on.
Genome stability related to cancer, there is always, in the back of my mind, and this comes back to Steve Jackson, right? I was exposed to a way of thinking, and I always had in the back of my mind that I should be thinking about the opportunities that may arise from our fundamental discovery work. And we published a paper in Science in 2009 describing a chromatin modeling of ASC1, which actually stands for amplified in liver cancer. And it turned out that this is a path-responsive enzyme. It's recruited very rapidly to DNA damage sites in a path-dependent manner. And we could show that this leads to local chromatin decompaction. It kind of opens the chromatin around the breakup and allows accessibility for other factors to come in. And so this was really interesting, but we started to have a think about why this was amplified into emerging cancer databases that were at the time. This was overexpressed in many cancers. And we did some experiments in animals to show that actually cancers that overexpress ALC1 become somewhat addicted to it. So somewhat like an oncogenic drug, and we didn't understand why that is. To be honest, we still don't fully understand it. But we were compelled that this was interesting. We approached Cancer Research UK Technology, as they are at the time, that now become Cancer Research Horizons. And we started a drug discovery effort with them, and we used the biochemical assay that we reported in the Science paper to develop a high throughput screen. We did lots of sort of chemical screens looking for small molecule inhibitors, and we started to develop them. This was kind of something on the side. It was kind of interesting. But then, I was approached by UC guys, and they were very interested in the possibilities.
Right, that is actually done for the day. Or until later on, 1 or 2.
If people have the opportunity to engage with venture, they really should. some of my best graduate students that go on to the